All right, we are going to hopefully finish off chapter 9 here. Again, continuing with metabolism, we're going to move into concept 9.5. Fermentation and anaerobic respiration enable cells to produce ATP without the use of oxygen. Now, most cellular respiration requires oxygen in order to produce ATP. Without it, the electron transport chain does not work, and this will cease to operate. In that case, we do have an alternative. If we take glycolysis and couple it with one of two processes, so either anaerobic respiration, which we cannot do, or fermentation, which we can do, we can produce keep producing a little trickle of ATP. So eukaryotic organisms can carry out fermentation, and we can carry out fermentation in a limited capacity, particularly when our muscles are under stress. Anaerobic respiration is limited to prokaryotic organisms. So we have different types of fermentation, and the whole point of fermentation is we are consuming NAD+, but since the electron transport chain isn't working, we would start to run out of this because normally it would dump its electrons onto the electron transport chain. So in order to regenerate the NAD+, so that we can keep glycolysis moving, we need to dump those electrons off onto something else. And typically we just dump it onto another organic molecule. There are two very common pathways: alcohol fermentation and lactic acid fermentation. As you can probably guess by the names, alcohol fermentation is carried out by yeast, and we carry out lactic acid fermentation. So this is what those processes look like. Again, you don't need to know them. You just need to know that the purpose of fermentation is to regenerate NAD+ when the electron transport chain is not functioning. So here we have pyruvate. So this is alcohol fermentation. Here we take pyruvate, we go ahead and pop off that carbon dioxide, similar to how we would do before, but instead of producing acetyl-CoA, we are just producing acetaldehyde. Okay, that we can then use as a place to dump those electrons that were put onto NAD through glycolysis, which produced pyruvate to begin with. We dump them back, dump it onto acetaldehyde that produces ethanol. So now we have NAD+, which we can cycle into the system, and you can get a very slow trickle of ATP, which is enough to keep some organisms alive and growing, such as yeast. Most of the time, yeast would prefer to do full oxidative usage, and they wouldn't want to do this, but they have this capacity. Us and other microorganisms do a slightly different deviation here. Instead of bleeding off that carbon dioxide, we just take pyruvate itself, use it as the terminal electron acceptor, and that will generate the NAD, and the outcome of that is something called lactate, which, as you can see with carboxylic acid group on the end, this is an acid, or otherwise known as lactic acid. But what we actually produce is the lactate molecule. Those protons are still out there. So again, just keep in mind that this is strictly to regenerate NAD. But we can use this to help us make things. So alcohol fermentation makes beer and wine. It is also part of the process that we use in making bread. Okay, it's not completely oxidative. There's a blend. We can also use this process to make biofuels. Okay, on this other end, on the acid production end, this is how we produce yogurt and cheese and vinegar. So in this, microorganisms are carrying out an acid fermentation cycle. The acid in these two cases interact with the milk and cause the milk to curdle, or it causes the proteins to denature and clump up, and that is what gives yogurt its thickness and cheese its chunkiness. Vinegar, that this is its acidity. There is things that use both of these processes. So kombucha and kefir both have alcoholic fermentation happening at the same time as lactic acid fermentation. Um, Consequently, in these, there might be a little bit of alcohol, but a lot of that alcohol actually gets consumed by bacteria that are also growing in there, producing acids. So you get a little bit of both. All right, so comparing fermentation with anaerobic and aerobic respiration, glycolysis, we get a net gain of 2 ATP, right? That's not a whole lot. In all three, NAD is oxidized, or NAD is reduced. It is the oxidizing agent, so it accepts the electrons during glycolysis. The process differs past that point. So in fermentation, we need to redump those electrons from NAD onto some whatever organic molecule in order to regenerate NAD+. In cellular respiration, we put that into an electron transport chain. This produces 32 ATP as opposed to fermentation, which produces 2 ATP per glucose molecule. There are bacteria out there that are called obligate anaerobes. They carry out either fermentation or some alternative anaerobic respiration process, and they cannot survive in the presence of oxygen. Oxygen is actually toxic to them. So this is an example of this would be when you dump those electrons onto sulfur to produce hydrogen sulfate, which is that rotting egg smell. Yeast and many bacteria are what we call facultative anaerobes, so they can survive in either scenario, right? So they prefer living in oxygen and utilizing that boost in power, but if they're without oxygen, they can still survive. Here, in facultative anaerobes, pyruvate is a fork, and you have a couple of alternatives, right? So here, we produce pyruvate. If there is no oxygen present, you would produce ethanol or lactate, depending on your mechanism. And if oxygen is present, you can then go ahead and send it into the mitochondria. And this is a ongoing decision that the microbes make. All right, the significance of glycolysis is in the ancient Earth, there was very little oxygen actually available. So this was likely one of the main processes, right? Glycolysis is an ancient process. It's one of the first biochemical pathways to develop. So this has been around a long time, and you can see this process happening in all range of organisms. Let's quickly go on and talk about concept 9.6. Glycolysis and citric acid cycle connect to many other metabolic pathways. And I touched on this earlier. So So these processes, these pathways, glycolysis feeding into the citric acid cycle, is a major intersection point with a whole slew of both catabolic and anabolic pathways. Again, this is breaking things down. Anabolic is building things up. So, catabolic pathways funnel those electrons into cellular respiration. Glycolysis itself can accept a wide range of carbohydrates. It doesn't have to be glucose. Things can be converted into intermediates along that pathway. Proteins must first be digested into amino acids. Those amino acids can either feed into glycolysis or feed into the citric acid cycle, and I'd pointed that out before. Consequently, these products from that pathway can also be used to make amino acids to build proteins. All right, fats are triglycerides are digested into glycerol, which can then go into glycolysis and into fatty acids. Fatty acids are used to generate a crap ton of acetyl-CoA. This is a process called beta oxidation. Consequently, you can produce a lot more acetyl-CoA from a fatty acid than you can from glucose. So an oxidized gram of fat produces way more than twice the amount of ATP as opposed to a gram of carbohydrate. So fat is a much more useful energy source. All right, so here we have proteins get broken down into amino acids, carbohydrates get broken down into simple sugars, fats get broken into glycerol and fatty acids. Here, glycerol, sugars, and amino acids might feed into this glycolysis cycle, so glucose to G3P to pyruvate. Here we have to pop off that amino group in order to get the amino acid. This will ultimately lead into acetyl-CoA. Other amino acids, there are parts of it which feed directly into here, and the fatty acid chains can feed directly into here. That ultimately leads into the citric acid cycle, which will then give you oxidative phosphorylation. Now, if you think about it this way, this is all a singular flow to produce energy. Well, sometimes we need proteins to grow, or we need fats to make phospholipid bilayer, or we need carbohydrates to build stored sugars or signaling molecules, or whatever it is we need to build. So this whole process is reversible. Okay, so instead of running forward, you can run it backwards and instead produce all of these things. Okay, this is why glycolysis and citric acid is something that we talk a lot about, because it is the central hub of all of these biochemical pathways. And if you look at a chart looking at all of these pathways, that becomes very obvious. Now, there are feedback mechanisms uh, in respiration. So, for example, if we have a lot of ATP, we would in fact inhibit a step of glycolysis, slowing this down, right? If we have excess citrate, this would also slow this process down. If we have excess AMP, it means that we have a deficiency of ATP that actually stimulates this pathway. So there are a lot of these feedback mechanisms, controlling steps along this pathway to help keep us in proper regulation. So when we need energy, we produce it. When we don't need energy, we can store it or build things with it.